Hello everyone, my name is Gregoryt Lebron. Today I will be presenting my research profiling and comparative analysis of secondary metabolites under variable fermentation conditions. Metabolism is the collection of all biochemical reactions in an organism. From these we got two major types of metabolites: primary, which are essential for survival, and secondary, which often serve ecological roles like defense, competition, and communication. Many important pharmaceuticals such as penicillin, streptomycin, and rifampicin are secondary metabolites produced by bacteria, especially from the genus Streptomyces, which is known for producing over two-thirds of known antibiotics. However, many bacteria contain silent biosynthetic gene clusters that remain inactive under typical lab conditions. That's where the OSMAC strategy comes in. By altering fermentation conditions, we can potentially awake these silent clusters and discover new compounds. These studies focus on four bacterial strains: Streptomyces flavivirens, Bacillus megatorium, Micromonospora aurantiaca, and Pseudomonas guinea. Each strain was grown under different fermentation conditions, with two types of media: triticioid broth and neutron broth, at 25 and 37 Celsius, and harvested at 24 and 72 hours. After fermentation, I extracted the secondary metabolites using ethyl acetate and analyzed the samples using liquid chromatography high-resolution mass spectrometry. The data was then processed with MZMine, and molecular networks were created in GMPs to visualize the relationship and clusters among the metabolites. As you could see in figure 1, the strain that responded most significantly was the Streptomyces flavorine. Next was Bacillium megatorians that also show significantly more features in triticioid broth at 37 Celsius. Meanwhile, Micromonospora and Pseudomonas show little to no change across conditions, suggesting that they may require more targeted stress conditions like iron limitation or co-culture. Now, as you could see in figure 2, GMPS molecular networking revealed distinct subclusters in flavorevents in triticioid broth at 37 Celsius. One cluster that is more watchable in figure 3, include methionine conjugated fatty acids, like to sulfur metabolism. These compounds were not present in nutrient broth, suggesting immediate specific activation of a critic biosynthetic gene cluster. Now, these findings confirm the hypothesis that fermentation parameters, especially media richness and temperature, affect secondary metabolite output in an strain-specific way. Streptomyces, being a prolific metabolite producer, responded strongly. Meanwhile, bacillus show moderate sensitivity, while Micromonospora and Pseudomonas were largely unresponsive under the tested conditions. This validates the OSMAC approach, when changing simple variables can unmask otherwise silent metabolic pathways. The structural annotation of sulfur-linked fatty acids in flavovirids under TSV suggests activation of previously unreported biosynthetic families, which may have a biological relevance. One limitation of the study is that chemical feature detection doesn't confirm biological activity. So the next step is to maybe perform a bioassay such as a disk diffusion to screen for antimicrobial effects. Also, future work should expand fermentation triggers like pH, salinity, metals, chelation, or co-culture, and confirm compound identity using NMR and MS fragmentation. Time course fermentation might also help determine when certain metabolites are produced. I'd like to thank Dr. Clark, Ria Gallant, and everyone at Holland College for their support and guidance. Thank you for listening.